So now let's learn detailed steps in eukaryotic transcription. Eukaryotic transcription is carried out in the nucleus of the cell by one of three RNA polymerases depending on the RNA being transcribed and proceeds in three sequential stages: initiation, elongation, and termination. Initiation of transcription in eukaryotes, unlike the prokaryotic RNA polymerase that can bind to DNA template in its own, eukaryotes require several other proteins called transcription factors to bind to the promoter region and then help recruit the appropriate polymerase. The completed assembly of transcription factors and RNA polymerase bind to the promoter, forming a transcription pre-initiation complex or PIC. The most extensively studied core promoter element in eukaryotes is the short DNA sequence known as the TATA box, Tata box, pronounced as Tata box. Found 25 to 30 base pairs upstream from the start site of transcription. Only about 10 to 15% of mammalian genes contain Tata boxes, while the rest contain other core promoter elements. So, the most extensively studied core promoter element in eukaryotes is a short sequence known as the Tata box. And only about 10 to 15% of mammalian genes contain Tata boxes, while the rest contain other core promoter elements. But the mechanisms by which transcription is initiated at promoters with Tata box is well characterized. So, recognition sites are there, promoter is there, transcribed region. Along with that, DNA, RNA polymerase, single-stranded template is formed. The Tata box as a core promoter element is the binding site for a transcription factor known as Tata binding protein or TAB, which is itself a subunit of another transcription factor, transcription factor 2B. After TF2D binds to the Tata box via the TBP, 5 more transcription factors and RNA polymerase combine around the Tata box in a series of stages and form a pre-initiation complex. One transcription factor 2H, TB2FH, is involved in separating opposing strands of double-stranded DNA to provide the DNA polymerase access to the single-stranded DNA template. However, only a low or basal rate of transcription is driven by the pre-initiation complex alone. Other proteins known as activators and repressors, along with any associated co-activators and co-repressors, are responsible for modulating transcription rate. Activator proteins increase the transcription rate and repressor proteins decrease the transcription rate. So, elongation. One strand of DNA, the template strand, acts as a template of 4 RNA polymerase. As it reads this template one base at a time, the polymerase builds an RNA molecule out of complementary nucleotides, making a chain that goes from 5' to 3'. The RNA transcript carries the same information as the non-template coding strand of DNA, but it contains the base uracil instead of thymine. RNA polymerase coding strand, RNA template strand DNA, coding strand, RNA template strand. RNA polymerase synthesizes an RNA transcript complementary to the DNA template strand in the 5' to 3' direction. It moves forward along the template strand in the 3' to 5' direction, opening the DNA double helix as it goes. The synthesized RNA only remains bound to the template strand for a short while. Then exists the polymerase as a dangling string, allowing the DNA to close back up and form a double helix. In this example, the sequences of the coding strand, template strand, and RNA transcript. Coding strand 5' to 3', template strand 3' to 5', RNA 5' to 3'. The dots indicate where nucleotides are missing. Nucleotides are still being added to RNA strand at its 3' end. Then termination. Sequences called terminators signal that the RNA transcript. Sequences called terminators signal that the RNA transcript is complete. Once they are transcribed, they cause the transcript to be released from the RNA polymerase. An example of a termination mechanism involves formation of hairpin in the RNA as shown below. Terminator RNA polymerase DNA. The terminator DNA encodes region of RNA that forms a hairpin structure followed by a string of eunucleotides. The hairpin structure in the transcript causes the RNA polymerase to stall. The eunucleotides that come after the hairpin form weak bonds with a nucleotide of the DNA template, allowing the transcript to separate from the template and ending transcription. Then eukaryotic DNA RNA modifications in bacteria. RNA transcripts can act as messenger RNAs right away. In eukaryotes, the transcript of a protein coding gene is called pre-mRNA and must go through extra processing before it can direct translation. Eukaryotic pre-mRNA must have their ends modified by addition of a 5' cap at the beginning and 3' poly A tail at the end. Many eukaryotic pre-mRNA undergo splicing. In this process, parts of the pre-mRNA called introns are chopped out and the remaining pieces called exons are stuck back together. 5' cap, poly A tail, exons, introns, splicing. Mature mRNA. Top of image, diagram of pre-mRNA with 5' cap and 3' poly A tail. The 5' cap is on the 5' end of pre-mRNA and is modified G-nucleotide. A poly A tail is the 3' end of the pre-mRNA and consists of a long string of nucleotides, only a few of which are shown. The pre-mRNA still contains both exons and introns along the length of the mRNA. There is an alternating pattern of exons and introns. Exon 1, intron 1, exon 2, intron 2, exon 3. Each consists of a stretch of RNA nucleotides. During splicing, the introns are removed from the pre-mRNA and exons are stuck together to form a mature mRNA. So, bottom of image, mature mRNA that does not contain the intron sequence. End modifications increase the stability of the mRNA while splicing gives the mRNA its correct sequence. If the introns are not removed, they will be translated along with the exons, producing a gibberish polypeptide protein, TATA, transcription start site, then TF, 2D, TBP plus 1 transcription start site. Eukaryotic transcription initiation, a generalized promoter of a gene transcribed by RNA polymerase II is shown. Transcription factors recognize the promoter. RNA polymerase II then binds and forms the transcription initiation complex. The 3 eukaryotic RNA polymerase RNAPs. The features of eukaryotic mRNA synthesis are markedly more complex than those of eukaryotes. Instead of a single polymerase comprising 5 subunits, the eukaryotes have 3 polymerases that are each made up of 10 subunits or more. Each eukaryotic polymerase also requires a distinct set of transcription factors to bring it to a DNA template. RNA polymerase 1 is located in the nucleolus, a specialized nuclear substrate in which ribosomal RNA is transcribed, processed and assembled into ribosomes. The rRNA molecules are considered structural rRNA because they have a cellular role but are not translated into protein. The rRNAs are components of the ribosome and are essential to the process of translation. RNA polymerase 1 synthesizes all of the rRNAs except for the 5S rRNA molecule. DNA polymerase 2 is located in the nucleus and synthesizes all protein-coding nuclear pre-mRNAs. Eukaryotic pre-mRNAs undergo extensive processing after transcription but before translation. RNA polymerase 2 is responsible for transcribing an overwhelming majority of eukaryotic genes, including all of the protein encoding genes, which ultimately are translated to proteins and genes for several types of regulatory rRNAs, including a microRNA and long coding RNAs, INC RNAs. RNA polymerase 3 is also located in the nucleus. This polymerase transcribes a variety of structural RNAs that include the 5S pre-rRNAs, transfer pre-rRNAs, pre-tRNAs, and small nuclear pre-RNAs. The tRNAs have a critical role in translation. They serve as the adapter molecule between the mRNA template and the growing polypeptide chain. Small nuclear RNAs have a variety of functions, including splicing, pre-mRNAs, and regulating transcription factors. Not all miRNAs are transcribed by RNA polymerase 2. RNA polymerase 3 transcribes some of them.